Hi, everyone, and welcome to the Genetics Podcast, where we explore some of the latest and greatest in genetics and genomics, and actually today in proteomics. And in this episode, I'm really excited to speak with Cindy Lawley, who's the Senior Director of Population Health at Olink Proteomics and the co-host of the Proteomics in Proximity podcast. Cindy works with many of the world's major population genomics programs like the UK Biobank, to name one, to bring together proteomics, genomics, and deep phenotyping using the Olink platform. So I've mentioned Olink probably half a dozen times on the platform, including one of the most recent in our 2022 roundup with Dr. Vera, who I got a ton of great feedback about. He was an amazing guest. We're going to try to get him back on for the end of 2023. And I've been wanting to do this podcast with Cindy for a long time. We met about a year ago at the Illumina. I think it was the first annual. I don't know if it's every year that it's happening, but they had this event.
Illumina Genomics Forum, I think they call it. IGF. Yeah.
IGF. And they had Obama, they had Bill Gates. I didn't get to meet either of them, but they were incredible speakers. But I did get to meet Cindy. And we bonded instantly because
I love the juxtaposition with Obama.
They had Cindy, they had Obama, they had Bill Gates. And yeah, we bonded over podcasting, but also Cindy, as you can probably already tell, is just a great person, super engaging. We got to talk about genomics, proteomics. So this is probably my most rambling intro yet. But Cindy, welcome to the podcast. Thank you so much for taking the time.
Thanks so much. Kind introduction. It's a pleasure to be here. I am a absolute fan.
I'm really excited about this. I've been excited for a while. You spent 14 years at Illumina, focused on genomics before you moved into proteomics. You spent some time at Metabolon and then now at Olink. I really want to hear about what spurred that change in a second. But first, I can't help but ask you, we're at Illumina for 14 years through probably one of the most exciting times in genomics, where the cost of sequencing went down probably more than tenfold. You started to see some major breakthroughs. What was that like, having a front seat to all that action at Illumina?
It was amazing. It was amazing. And it's only in retrospect that you realize what an amazing time it was in history, right? And so, when I look back, I think, there were times when I knew that I don't know, the world was making history. It wasn't Illumina necessarily. It was what people were doing with that technology. But just looking back, I treasure that the luck involved in me getting to have a seat there, and the people and the relationships that I forged from that experience will be relationships that I'll have for a lifetime. I treasure that. And I think about, if someone else had interviewed me, I was straight out of my PhD. If someone else had interviewed me other than John Stolpnagel, would I have even considered going to an industry position? I really was not planning on taking that role when I interviewed for it. I really thought academia was my path, that teaching was my future. And so it was just a bit of a surprise coming out of talking to him that I was so compelled by the technology. And I'll tell you, that interview, I maybe understood 30% of what he was explaining to me. He was, I had to hear it maybe 3 times before I really got the scope of what was possible. This was pre-sequencing, right? This was array genotyping when I joined. And so, had no idea that Illumina was going to be the powerhouse it became back in 2007 when Solexa merged with Illumina. Yeah, that Solexa technology and the people behind it are phenomenal.
What did you do your PhD in? Was it something related?
Yeah, so it was evolutionary biology, and I had done a master's first, an independent master's at another university in cladistics and evolutionary biology, and found that I really wanted to study something in my PhD that had an applied, that was had a reason. I didn't want to. I wanted it to have a solution for something, and so I ended up using genetics as tools for measuring fish populations and understanding the fisheries. So ultimately, my PhD was under the umbrella of marine science, but it was funded through the NOAA, National Oceanic and Atmospheric Administration, where I worked as a contractor and as a student throughout my PhD work.
In that field, fast forward to today at the Sanger Institute down the street from where I am, they're doing the Tree of Life project, sequencing every animal species they can basically get their hands on. It's pretty amazing what 20 years or so difference makes.
Yeah, and the cover of Science, Eleanor Carlson from the Broad Institute, who I don't know, I think pretty much everyone knows, she'll have the cover. She just texted me the cover. It's going to be an amazing cover on Science. And again, the just an understanding of comparative genomics and what we're learning from it. It's the path not taken, for me now, but it's near and dear to my heart since I'm now so focused on the human arena.
Yeah. That's maybe a good time to transition to talking about your shift from genomics into proteomics. Although I think Olink is genomics in a way, and we're going to get into the technology,
Mm.
but or sequencing at least. You shifted from Illumina into Metabolon and now Olink. What spurred that change? What made you decide after 14 years to try something different?
Yeah, good question. So Illumina had grown from about 150 people when I joined in 2004, and again, it was just an array technology company, right? Our biggest competitor was Affymetrix, and I will say that that time in Illumina reminds me a lot of this proteomic space right now because the targeted approach to looking at the proteome is really what's driving high throughput capabilities. That these affinity methods that can do high numbers of samples are targeted. They're not measuring everything that's possible in the proteome. They're looking for known assays that are developed and
Mm.
qualified as specific. So that array genotyping, that approach of trying to get it to as much information in the genome as you can, we're trying to do the same thing with the proteome, try to get as big of a breadth of, cast a wide net, we say,
Yes.
to fuel discovery. And we can talk about what I think is innovative about Olink and the reason that I came to Olink. But the transition out of Illumina was really because I had led the consortia program at Illumina. Once the sequencing technology was on board, we had the two sides of our business, and sequencing was a logical way to drive discovery of SNPs in different populations and catalog that variation and deploy it in an intentional way, a tag SNP selection way on arrays and make access to the genotyping data cheaper. And it's gotten cheaper and cheaper over time, right? And still people sequence where they can afford it, but the genotyping capabilities at Illumina had continued and still continue to improve in pricing and coverage, right? It's moving in two dimensions. Just like in the proteomics, we're trying to increase our coverage, but drive down costs to make it more accessible. So that program really wasn't needed by about 2014. I stayed for another 4 years and moved into a market development role and really drove aspects of the business in South America, in Asia. And I loved that time. But the reorgs within Illumina gave me an opportunity to exit and take some time off. And I laughed at the time because I hadn't had a summer off since I was like in junior high.
Yeah. What did you do? What did you do with your time off?
I got a puppy. I got a
Nice.
golden retriever puppy,
Amazing.
and yeah, isn't that funny? Yeah, so I took some time off, and then the transition into Metabolon. I will say it was a little scary taking time off because everybody was telling me, if you're leaving a job, the best time to get a new job is when you've already got a job, and like maybe that's true, but I just thought this gives me some time to really explore different companies, what they're doing. The two things that were highest priority for me are culture and cool technology. And I was looking for technologies that were a rising tide that lifted all boats because I had become really enamored with that with the Illumina technology.
Yeah.
And so I took the time and I attended conferences. I attended AGBT. I did some contracting work. So someone paid to have me attend some conferences, which was really great for me too. And I just started making a list of companies and contacts I had in those companies and what I liked about them and what I thought about the culture. I joined Metabolon because the role was in population health, and this was akin to my days in the consortia program. And that was some of the funnest accelerating of discoveries that I'd ever experienced anywhere in my career. And so I was trying to kind of find something that recreated that opportunity because I felt like my personality is a bit deep into the science, but also very, very social. I don't know
Yeah.
if that's the right word, but but someone who can bring collaboration and accelerate collaboration. And so that population health role at Metabolon was attractive to me. And I really am all about the manager, right? So Alex Forrest Hay, fantastic leader, I think. And so I worked for him for about a year and a half until he left Metabolon. I continued working at Metabolon until I saw, and we can talk about metabolomics. Certainly they're the currency with which the microbiome pays us to house them. That's how I think about metabolites, is they measure our, an understanding of what's going on in interactions with the microbiome. There's some interesting papers that have come out with proteomics, metabolomics, as well as genetic data. But so I saw the launch in 2020 of the NGS readout that Olink had. And I had some friends over at Olink. And so I reached out to Olink and said, this is so cool. And it really will solve a problem that we have been waiting to solve, to get closer to the phenotype, but in a broad scale way. I'd really like to be part of it. And this is how I think my background might be beneficial. And I got in front of the right people that agreed. And we put together this structure to work with the population health space to help folks that are working in genomics or come from a genomics area, might be trying to get at real-time biology using RNA sequencing, which is a great way to dig into that might have additional value by adding proteomics.
So tell me about proteomics, because I actually know very little about proteomics except for what I've learned from your podcast. I'd love if you could just paint the landscape. And I loved actually the metaphor of arrays compared to maybe whole genome sequencing, because I do know that there are different technologies that measure it in different ways, but I'd love if you could actually just give me a crash course in proteomics.
Yeah, sure. So this is one of my favorite things. So I usually talk about the plasma proteome because that's sort of the liquid biopsy space. That's a low invasive sampling approach, we say, right? Taking a blood draw is a lot easier than going in and say biopsying a tumor or some other tissue if disease is present. So just in talking about the plasma proteome, mass spec methods are the gold standard, really. I think most people would agree that in theory, mass spec captures everything that's there. Mass spec is a hard technology to do thousands of samples through. It's possible, but it can tie up the instruments for a long time, or you need a lot of instruments. And frankly, mass spec does the best job, and I am not a mass spec expert, so please forgive me if I state this incorrectly. Well, I think some amazing discoveries like HDL and LDL are clinical labs we all have access to when we go to Quest or LabCorp to have our health monitored. Those discoveries are well suited and driven by the kinds of things that mass spec does really well, because they're high abundant. So meaning they're high presence. There's a lot of them in our blood. And so they come through on the mass spec instrument very clearly, and they're quantitative, and those correlations are really clear. What is more challenging to do on a mass spec instrument is to look at the low abundant proteome. So these cytokines and chemokines became really talked about in COVID. Those are not as easy to look at, especially in large numbers of samples. And so what I think the Olink technology does really well is concentrates the library on these low abundant proteins. And then over time, we've expanded our library to also be complementary and overlap to some extent with mass spec. So we, and that protein list you can download from our website without even putting your email address in. So that, we're pretty open about what we've got on our assay, and we welcome contributions or requests coming from researchers on what we should add. Okay, so that's where I think Olink is special. The way the assay works is it's two antibodies. So antibodies are familiar. We use antibodies in drugs, right? So antibodies hook these proteins out of solution. It's thinking of plasma, right? And they have conjugated to these antibodies, these single-stranded oligos that can then hybridize once both antibodies find the target protein. Once that hybridization happens, you're just converting it to a library prep, right? So once that hybridization happens, you can extend it, amplify it, and it will be a counted reflection of the original protein levels in the original sample. It's a relative concentration. But the readout can be on a qPCR instrument, which was our original deployment of the technology that maxed at our target 96 panels. So we have 92 unique proteins on each of those panels. And so people would run multiple panels to get as much discovery as they could. Now we've got the 3000, the Explore 3072 product, which is deployed on an NGS sequencer. And so we can do more coverage. We can do 384 samples at a time for those proteins, so around 3,000 proteins in one run of a NovaSeq today. And then, of course, the sequencing costs keep getting driven down by players, not just Illumina, but some of these other players on the market like Element and Ultima and MGI and Singular, right? These are ones that we are quite open to working with as well. We've had press releases around working with three of those from AGBT. Yeah, so we're keen to see the cost of the sequencing component be driven down so that the cost per sample can be driven down. And then we're also trying to expand to cover more of the proteome because these discoveries are so ripe. We've got like 1,200. So April 15th, Gary sent out our latest publications list. So 1,224 peer-reviewed publications just since 2016. So that is pretty phenomenal. And seeing that makes a big impact on me when I look. And that's just the peer-reviewed stuff, right? We've got other stuff in bioRxiv, like the UK Biobank Pharma Proteomics project that preprints out. Came out last year, and we're waiting for the peer-reviewed version. That was a long answer. I hope that was.
No, it's great. It makes total sense. And I guess it seems like you could compare it to an exome or an array or something like that in the sense that you're building a big panel.
Exactly.
And ever expanding, and you don't see what you don't see, but you can do an incredibly high throughput dive into some of these low abundant proteins that would be very difficult to see from a lot of other methods.
Yeah, and Karsten Suri and Jochen Swank and Mark McCarthy have published a paper to say, what's the value of adding this intermediate phenotype, this protein level information when you've got genetic information and you've got clinical data or phenotypic data. The power that you can have to amplify your ability to detect what's going on between the genetics and the disease is much higher. So you're increasing power. Karsten says it's like a magnifying glass that allows you to do what you need to do in smaller sample sizes. So I think that's also exciting as well. I think, yeah. So yeah, I'll just stop there. And yeah, I'd love to talk more about the UKB Pharma Proteomics project as well, just because what they're doing there, I think, is really innovative, and those data will be publicly available ultimately. Some of it already is.
Yeah, it'd be great to hear about that actually, because I think that's one of the best examples of layering. We know most listeners will know that UK Biobank has started with sample collection and phenotyping, layered on genotype arrays, then exomes, now whole genomes, a couple of different types of proteomics as well. And there've been a couple of papers that over the last year or so. It'd be great to hear more about that and what that is, what you're looking for.
Yeah, so many of your listeners will know that just to use the Exome Sequencing Consortium as an example, that was a pre-competitive collaboration. That's a term that we throw around, meaning that it was partners from industry that came together and funded this discovery data set that they knew would be publicly available and is now. So that ability to drive forward something with funding from multiple partners, it accelerates innovation, right? It accelerates the ability to have that sequencing data in the community, and it's very exciting. And so on the heels of that Exome Sequencing Consortium, it was proposed that a similar effort be organized around collecting proteomic data and doing a broad proteomic look at that time. So some of those ESC partners stuck around for that effort. Other new partners joined. And ultimately, we have 13 pharma partners that were part of that UK Biobank Pharma Proteomics project, which is what we call it, UKB PPP. So that project made the choice about technology, chose Olink for this particular effort that was originally around 55,000 samples. It's now expanded with a COVID project to over 60,000 samples. And they first committed to doing our first product, which was the Explore 1536. So it's around 1,500 proteins. Those data, that first tranche of data, are now publicly available. I confirmed with Naomi that they're on the UKB research analysis platform that's hosted by DNA Nexus. So those data should be publicly available. The second tranche of data, the additional expansion to around 3,000 proteins, will be coming out later on that platform. And so just this ability to have pharma come in and fund it demonstrates, I think, that they've got some combined interest, but also, of course, individual interest in the ability to look broadly at proteomics. So the publication looked at, and this is a preprint publication led by Ben Sun and Chris Whelan. Ben is at Biogen, Chris is now at Janssen. That preprint identifies 10,248 associations, correlations between genetic regions that are known from the UKB data with protein levels. So sort of a dosage effect going on with the genotyping. You imagine that these are significant correlations. 85% they report are novel. So I think this will provide a beautiful data set with which to start assessing with co-localization or Mendelian randomization these methods to look at the clinical data and which of these might not be happening by chance alone, to then distill it to a subset so that there's a list of potentially causal relationships between genetics, proteins, and disease. That's the magic, I think, of having those three data sets together in this place, in this research analysis platform. That ability to have a more systematic approach to evaluating these potential therapeutic targets that's disease agnostic and allows each pharma to rank these potential targets in terms of what their priorities are. I think that has some promise. And I think it's pretty well established that when you have genetic data going into a clinical trial, just speaking the language of the pharma here, that you have more than twice the likelihood of it being a successful clinical trial, right? We don't know what proteins will provide for clinical trials, but I would imagine it's going to add additional value as well. And so I'm excited to see how that pans out. It's early days at the moment, but we've certainly seen some exciting publications around post-clinical trial analyses that give mechanistic insight into what's happening with a therapy and allows pharma to have a biomarker or a set of biomarkers that are helping with better diagnostics. So that helps them have a population that's appropriate for prescribing that drug, as these have to be moved to the clinic, of course. Having a biomarker for diagnosis, having biomarker for tracking disease progression, and having a biomarker that helps with understanding response to treatment, right? That's a triple threat that I think adds a lot of value to them. So we've got some nice examples of that, which it's always wonderful when pharma can publish on their results. Of course, so much of what they do, they need to do internally. So I can give you some of those links to some of those publications for the show notes if you think.
Yeah, that'd be great.
It's phenomenal, I think, integration of multi-omics.
Yeah, and I think to go back to the very first paradigm you explained, I think this is such an important one, because if I could play it back, we currently live in a world where we've got really strong links between genetics and all kinds of if we just restrict to the medical world, genetics and medical phenotypes. We know that LRRK2 is strongly associated with Parkinson's. We know that ApoE4 is strongly associated with Alzheimer's. By layering in proteomics in between those two, then all of a sudden you get a lot of really interesting data and potential stories to chase down, where you can look at the genetic link to the phenotype via the lens of the protein and look at, okay, the change in ApoE4, we know and suspect that it changes the ApoE4 protein, but what else does it change? And does that give you a different potential target to go after, or to the point you raised, maybe there's another biomarker as well that's easier to measure or more sensitive or flips on earlier
or druggable. Yeah, and I would even say, these really highly penetrant or strong associations, I should say, strong associations like the ones that you describe are not the ones I think we can add the most value to. Although, that's maybe because I work in the polygenic world quite often. I'm talking to people that are looking at diseases that are quite, that they're building polygenic risk scores that might have 100 or 1,000 different genes involved. And in fact, now when I'm at ASHG or I'm at a meeting where someone's talking about polygenic risk scores and they put up a gene list, I'll take a picture of it. I'll put it down next to me. I'll go into our Insight browser. I'll make a list of those genes and I'll see how many of those proteins, which are the products of those genes, how many of those proteins we have in our panel to see, what might be the value that proteins might provide and understanding sort of the mechanism there. And this is aspirational, but that polygenic aspect means that you need a polyprotein, a multi-analyte solution. And I think the technology having originated as a qPCR readout in a midplex size, we have target 48, which has 45 proteins in it. We have a target 96 that has 92 unique proteins in it. We have redundancy for QC, et cetera. But the ability to take the discovery and cast this broad net and then narrow it to a protein panel, especially with these AI methods. Of course, you can't have a podcast without saying artificial intelligence.
Not anymore.
Right? But these approaches to have a sort of maybe even the different nodes in the biological pathways that are involved and represent each biological pathway in your signature. But you can imagine, these polyprotein signatures. It looks like 20 proteins in many cases can be very impactful, 20 proteins or less. In fact, there's a preprint out about, I think it's 12 different cancers that Matthias Uhlin has looked at, and his team have looked at for characterizing different cancers. So these subsets, and they have it down to, in some cases, fewer than 15 proteins, right? So being able to have these specific assays that measure the protein we say we're measuring allows researchers to drill into the actual pathways and trust that they'll validate on the back end. And that's essential to be able to demonstrate clinical utility. Again, we're early days, right? But that path to an LDT, we've got several customers that are on that path to an LDT for building signatures that help with, Crohn's and colitis, predicting which pediatric cases will develop complications, which ones may have a time period where you can insert a pause before you take out a kid's colon, having an ability to score the likelihood that you're going to have those complications. It's pretty valuable. These cancer risk, we've got ovarian cancer, Crohn's and colitis, multiple sclerosis, we've got a dozen different Alzheimer's, neurodegenerative disease, right? Big area. These areas where folks are really focusing attention to build these predictive tools. And then, of course, aging is such an exciting arena, right? So lots of interest in trying to build predictors as to who will age with less cognitive decline than the rest of us. So we want to, wouldn't it be great for medical care to be predictive like that to help us monitor things in a more personalized way? That's the dream, right?
Yes. You just in the last 10 minutes, I think you've named six or seven amazing applications of it. I'm curious whether there's one over the last couple of years that has really captured your imagination. Is there an application of proteomics that has just made you see the potential in a unique way or have an aha moment of some sort?
Yeah, so I always lead with it, right? So it's the utility of this in the context of what I've invested my career in, which is genomics, right? How do we empower the genomics with the proteomics? So you'll see that phrase in our messaging quite a bit because we see that the genomic data sets are powerful, and if we can add value to those, that's something we're excited to see. And I think we're seeing that in many cases. So that's, I think, definitely this ability for pharma to have a more systematic approach to identifying their targets is probably the most exciting area for me, because I want us to have more successful clinical trials. I want us to have tools that will help us understand the mechanism by which a drug is acting. I mean, how many drugs are on the market that they're fewer today than there were 20 years ago, but they get approved and they go through, and we don't actually quite understand how they worked. And I think we're starting to reveal that. And I think also this liquid biopsy space is such a hot space, right? Cell-free DNA and the ability to do early cancer detection is phenomenal, but it's a burden to have false positives. It's a burden emotionally for someone getting that result, right? And
Yep.
it's a burden to the healthcare system to be monitoring someone who is not actually having cancer. And so, is there a way that we can add value there to improve the accuracy of those tests? And I think from what I'm seeing, I think that there absolutely is. So we'll see, but it's exciting. I'm having the time of my life.
That's amazing. I was searching for proteins on your website in the background. I have a particular protein that I'm really interested right now that I was able to find. I don't think I could say it on air yet, but I will be asking you about it afterwards.
I will pull up any paper we have that has a hit on that protein. So we actually have, this is a great time for me to actually point out a free tool that we have launched in the last year called Insight, which is it's just insight.olink.com. Very simple. You can make an account. You'll need a username and password because you'll want to save protein lists. And this is where I save protein lists and I share them with people that give talks about polygenic risk scores who might think I'm crazy to send them this list of proteins. But I like people thinking about the future. So yeah, that interface is beautiful because you can actually browse pathways, but you also can put in a protein and see any publications in our database that reference that protein. You can see which panels that
Oh,
protein's on,
cool.
and you can, mostly it's a list of proteins, right? So any I looked up inflammaging, right? Inflammatory aging and cognitive decline and Alzheimer's disease, and for a client, and came up with a list of proteins so that she could try and determine which of our panels should she do a with her budget. Should she do a target panel, or can we get her onto an explore, more of a discovery tool? We have multiple panels in our explore, so it's modular. We have these 384 protein subpanels within the 3072, so someone can choose to run inflammation markers and just run a subset. So that allows us the flexibility to help hone in on what they might be interested in or where they think that their signal might be. So that's also useful. So I think that the tool can be fun to browse, even if you're just trying to understand pathways.
Yeah.
And also, you asked earlier about proteins, and it's probably worth mentioning that you can have abundance, sort of an average abundance of any given protein, but you imagine that when you plot that in picograms per mil or femtograms per mil or whatever, there's going to be these little whiskers up and down on that protein. And so unlike genetics, you need to hone in on that dynamic range for that given protein. And this, I think, is the magic of the assay, and the way that our assay is set up is really important to keep in mind to test an assay against both diseased as well as healthy samples, so that you can see in a case control study for like a biomarker study. Many people have done biomarker studies without genetics on our platform. And so comparing those cases and controls, you want to make sure that you're able to see the difference in those. So capturing that dynamic range and the readout of, whether it's on our qPCR instrument or NGS readout, that's an important aspect that you don't have to. There's some comparisons you can make to RNA sequencing, but our team doesn't like it when I compare RNA sequencing to proteins. They're a little different, but which
Yeah.
I understand. But that's where I come from, so I think about that.
Right. A question I should have asked maybe at the very start, but how many proteins are there? I assume there are at least 20,000 because there are at least 20,000 genes, but we've got different isoforms. How does all of that work?
Yeah, so I've heard estimates around this that we can assume 20,000 proteins from 20,000 genes. Those are the full length proteins, right? So then you've got glycosylated proteins or phosphorylated proteins. These sort of proteoforms that are ripe for an understanding of discovery. And so I think an estimate that I've kind of taken on is that there's probably around 100,000 total, maybe, of including the proteins as well as the various proteoforms. But I don't think we know. I think this is this is an area for, in the future, when and if we're able to actually sequence the proteome and see everything that's there and be able to do it in a high number of samples so that we can understand diversity across different populations and how they express proteins. And I think, that's another area that I think we're pretty passionate about is to have the proteomic data in the world be representative of underrepresented populations in the genetic space. And so if we can get ahead of that now and see how we can enable an understanding of proteins in different ancestries, I think that's going to be really interesting. I mean, you can think about it as exposures over a lifetime, right? If I've got a genetic variant that someone else doesn't have, that changes my protein levels, that protein level is going to be an exposure I'm going to have over a lifetime that other person might not have. And we absolutely know that there are differences across ancestries, just like there are in genetics, right? Of course.
Yes, I was interested in along the theme of exposure around longitudinal sampling of proteins. Have there been any interesting applications in any of the major biobanks or otherwise of
Yeah.
taking measurements over time? Because it's not something that we need to worry about with genetics, but that's one of the downfalls of genetics. It is a single static measurement your whole life, with the exception of somatic testing.
Yeah. And I think, some people would say methylation is one of the attempts to get at
Yeah,
more real-time biology. RNA sequencing is an attempt to get at more real-time biology in the genetic space, right? Using piggybacking on the cost reduction in that space. So there have been longitudinal studies that have been done on the same individual over time, and it's surprisingly constant in one individual. So that I think there's some changes that can happen at certain life transitions. A woman having a baby, going into menopause would change proteins. Just speaking on my gender. So these things will shift some proteins quite dramatically for a period of time. But as I understand it, and again, this is representing other people's research, but it's a bit surprising how over time, how consistent it is over time. I think I'll also mention the opportunity for being able to see deviations from a background of a control population is also quite powerful. So there's a beautiful study that I talk about quite a bit that came out of Aravale cohort. That's the Institute for Systems Biology. So Aravale was a business that was trying to that did build a cohort of about 5,000 people and monitor them over, I think every 6 months they tried to get blood samples, and they biobanked these plasma samples and have maintained them.
wow,
Even though Aravale closed its doors, those same samples are still there and they're still generating discoveries. And they still maintain the institutional review board for this cohort. They're still able to reach back out to these folks. So the consenting there is really quite beautiful as well. And it was originally a coaching program to help individuals with their health. And over time, it's been just this ripe source of information because ISB is, they do microbiome, they do proteins,
right.
they do metabolites, right? They're rich systems biology analysis experts in my view. And so they had these samples that over time, some subset of individuals developed cancer, different cancers. And so they did a study and they got this into scientific reports with a pretty small sample size, but longitudinal sampling, where they had 10 individuals that had developed cancer, where they had at least 3 biobanked samples prior to diagnosis. So then they ran the proteomics
Oh,
on those, retrospectively, and looked for signatures relative to, I think their control population was 69 individuals. I think it was originally 70 individuals, but one of the individuals in the control population looked like they might have been out of, they might also be needing a diagnosis. So it was,
right.
and they included that in the paper, which I found really interesting. They weren't
Wow.
able to to get that person to respond back, but they talk about it in the paper, which I think was really interesting. But what was really exciting out of that paper was a tissue agnostic signature, a protein that looked like it was indicative of metastases across different cancers. So I think the cancers were lung, breast, and what's the one that Steve Jobs had?
Pancreatic.
Thank you. Pancreatic. Yep. Yep. So those across those three, so they say tissue agnostic, which just means across different cancers, right? But those were the three cancers out of the 10 that had shown metastases. And so,
Huh.
so that idea that there was a marker that was a signature across cancers for metastasis was pretty fascinating. A preliminary study, so small number of samples needs to be validated, but that kind of thing is possible
Amazing.
with these longitudinal cohorts, right? And the investment that's been made in maintaining these samples and limiting the what we call pre-analytical variation, trying to make sure that the samples were collected in as consistent a manner as possible, and that how they're collected is tracked, and all of those things go into an analysis like this to make sure they're not confounding results. And so it's an exciting paper. I can provide the link to that as well.
Yeah, that would be great. We've talked about the consortium projects that you've been a part of and helped lead over the years. These are it's really difficult to get any big group of people to do anything, but it's extremely difficult to get a big group of people that work for big companies to come together and do anything. But it strikes me that you have a little bit of a talent for this, or at least a passion for it, and it's not easy to do. What have those experiences been like, and what have you learned about trying to get large groups of large companies together towards a common mission that they all really want to be on, but sometimes the structures can just make it really difficult to get everybody rowing in the same direction, to use a rowing metaphor. What have you learned about that?
Yeah, so I certainly can't take credit for having gotten this UK Biobank Pharma Proteomic. I just want to make that really clear. That was Chris Whelan, Lyndon Mitnall, and Melissa McCarthy, Melissa Miller. Oh, Melissa McCarthy, isn't she an actress?
Yes,
Clearly.
yes, a good
Yeah.
one. Yeah.
A really good, really funny one. Yeah. Melissa Miller. So sorry. Yeah. Melissa is at Pfizer. Chris is now at Janssen, and Lyndon is at Regeneron. So those three really spearheaded this. And what Chris says is that what was important there was to provide a really clear messaging that everyone agreed on, who were science focused. And these are scientists within industry. So
Right.
that's a really important aspect, is that if we look at pharma as a funding body, that's not really going to be a successful approach. These are very heavy hitting scientists that we're talking about, who are driving this experimental design and
Right.
proposing to the UK Biobank to use the samples. And so having really clear messaging that all the companies could then take internally with advocates and senior leadership to drive that discussion was really important. And not everyone could move forward in that partnership. Some people just couldn't get that in the timing that was needed to move it forward. And so that will happen. I still think it's amazing that there were 13 pharma partners that are in now, and I think there's potential. Who knows what's next, to potentially have additional partners come in or go out. We'll see what Chris is able to do, or what the other partners are able to put together. So that I think was critical. The aspects of having a really clear guideline around what's possible, what's shareable, what's not shareable, and what the goals for success would be, so that it can be pitched internally to each of these companies. And then I think there's a certain element of a fear of missing out that probably comes into each of these companies. Although I can't speak for how those conversations advance, but I would imagine, not wanting to be left out and not have access to such a valuable, powerful data set. And I just mean, the UK Biobank, Exome
Yes.
Sequencing Consortium, the proteomics data, right? These, not just this data set, but multiple different endeavors that were pre-competitive, I think must play somewhat of a part. And then I think the scientists having good communication with their legal teams, so that their legal teams understand what's a risk and what's not a risk in terms of the science, what's publishable, what needs to be kept
Right.
proprietary. I think that's got to be senior leadership, legal, as well as the lead scientists from the joint steering committee for that UKB PPP. And then just back to my consortia days, when I was at Illumina, I would say that what helped a lot for me was really the recognition that SNPs were probably not going to be patentable.
Yeah.
And so Illumina's ability to see that there just wasn't likely to be, that this was something that would help the participants as well as help Illumina. And that we were trying to, the way that Illumina manufactured its arrays, and mostly these were consortia around getting access to sequencing data so that the group could build the best SNP list to drive forward an array that was going to help in whatever field. So maybe it was psychiatric disease. A lot of these were agriculture arrays. So in fact, the dairy industry has been completely transformed in the way that they choose their producers by the tools that came out of one of those consortia. So that's one of my most proud participatory events at Illumina was going to receive USDA awards around that, as well as building a pig and sheep chips. Those agriculture scientists are so scrappy and can do so much with
Right.
their budgets, and brilliant in the informatics, right? Because nobody's making canned software tools to help them analyze their data. They all are doing it. And so, a lot of my time in my last few years at Illumina was about evangelizing what some customers were doing with other customers, and just making connections, which
Right.
is just so much fun. It's a lot of fun.
I was curious about the trajectory of proteomics as a whole. So you were had a front row seat to just the most amazing Moore's law on steroids with genomics over 14 years. Many of us have seen the graph. If you haven't, then I'll put a link in the show notes, but just Google.
It was just in The Economist. It was just in The Economist last week. My mother actually saved it for me, and I was like, yeah, I know that graph.
Yeah, I've seen that graph in 50% of all presentations over the last
That's
10
right.
years. But the drop in, you mentioned earlier, drop in not just price, but also throughput efficiency has been astronomical. Where are we at with proteomics? What does what does a proteome cost today? I know we've talked about the throughput quite a bit. What did it cost 5 years ago? Where is it going? It'd be really interesting to learn a little bit about that trajectory.
Yeah, great question. So we were just trying to sketch this out for a graph that, and I will say there was a point where I promised myself I was no longer going to show that NHGRI graph.
It's too good.
It's too good. You have to show it. I remember getting the bullets from Jay Flatley around, $3 billion, 13 years. For those were the numbers we used, the public funding, right? And then the cost at the time he was quoting it was like $1,000 in a day, right? So just making that comparison, it allowed me to talk about genetics with my family and help them understand why I was excited about it. And anyway, back to your question, where are we at with proteomics? So when I started at Olink, we were a service organization. So that's very much the way Illumina launched the array platform as well. That the array technology was a service business. And then as, first we built the big genome center array technology platform, right? That went out to the big 5 genome centers. And then we built the bead array reader that was more, for more broad application, different kinds of, not the big institutions, but the smaller core labs could run that array technology. But in the early days, we were running projects internally, and it was around $1,000 a sample. I mean, these were expensive genotyping projects when they were doing custom SNP discovery, right? I would go to dbSNP, pull all the SNPs in this gene list that they had, submit it back to the customer. I was a project manager, and help them pick their SNPs for the manufacturing. We would make the manufacturing of that, and that we would make them in big vats, right? So the economy of scale was really clear in the Illumina technology, that if you could bring more people together, you could really bring down that cost, because you could leverage this pool. Because to kick off a manufacturing event, you had to make a lot. Anyway, so those projects were quite expensive. When I came to Olink, service organization, and so all of those service costs were rolled into it, and it was around $1,000 a sample for the broadest look, that NGS. And at that time, I joined, as I mentioned, after the NGS launched. That first product was called the Explorer 1536. And so that was about what we were charging there. As we've expanded the coverage of the proteome, we've tried not to have the pricing go too much higher, right? Because you want to be driving it down. And so, so the pricing today, I think, essentially we're able to get at the best pricing if someone's running it themselves. So our business model is really, as people have gotten more confident in the technology, they want to bring it in-house. Decode Genetics is a company that probably would, or a group within Amgen that probably wouldn't be running Olink if they weren't able to have it, their hands on it themselves. They love that, and they tweak it, and they're so valuable. Their feedback is so magical to help us improve our products. But yeah, so our business model is now moving it out, and we've got lots of these Explore labs all over the world today. So that's the cheapest way to do it. And I gave pricing when I was at AGBT, ballpark, it's not the lowest pricing that's possible. It's not the highest pricing as possible, but it incorporates the sequencing component, which I think we estimate is around $63 at list price today on the NovaSeq 6000. So that has to be factored in. But I think we can get around, 300, 350 pretty routinely, right? So, but these are volumes, right? So we're very much like arrays. The more you order, the more you can. So I'm talking about driving down costs with big orders. And then when groups are doing something like the UK Biobank, we got to figure out, what is their budget, what's possible, and try to do the best that we can to enable innovation, because it's going to be seen by everyone in the world. And so that's sort of off of our little tiered structure. So, but I think it's a reasonable number to have in mind.
Coming down the cost curve dramatically, it sounds like.
Exactly. So, and then if someone is running it as a service, there's going to be some costs associated with their service FTE, right? They're people that need to run the assay. So I don't want to set the expectation that if you're accessing it from one of our service providers, that's going to be the price. I'm saying if you're running it yourself in your lab, you can get it down to those prices. So I used to say a fraction of whole genome sequencing, but it's not. If we're getting whole genome sequencing down to $100 a sample, right? We're moving in that direction, which is super exciting. And it'll drive down the cost of this too.
Yeah. And nobody's giving $100 whole genome anyways. I'm very confident that it's coming, but like you said, you've got service on top of that. And any of the medical people will be very quick to tell you that the analysis and the genetic counseling time and the doctor time costs very much on top of that.
Well, and we gotta say then that this is research use only, right? So we are not a clinical company. We're not providing clinical services. However, we do have customers that have made the discoveries that they're now working with us to deploy on these midplex. I think Octave Bioscience is a pretty well-known one because they've done these webinars on all of the steps that it takes to validate, and we can do an exact quant tool that they can then demonstrate clinical utility and have an LDT for. But that's theirs, right? That's us in the background supporting it. But I don't want to set the expectation that we're building clinical tools today. But I certainly see this as a path for translating what we've learned in the research field, and our customers. What I love is having our customers take this on and carry it to the clinic. And like I said, early days, but that's my aspiration. That's my hope.
You mentioned an LDT there, just for people who are new to that. It's a laboratory developed test, right? Is what you mean. It's basically
right. Exactly. Yeah.
A baby in vitro, instead of a fully FDA approved in vitro diagnostic. If you're running this in one lab, you've got really strong analytical validity. You can run it as what's called a laboratory developed test, right? Have I got that?
Yeah, that's right. And it's tied to that lab. It's very specific to a lab, which is hence the name, right? So, and again, I will say I'm not an expert. I think you're probably learning I'm not an expert on anything, but I touch on a lot of different things, and I'm fascinated by them. So yeah, I'm not a clinical specialist, but
I think you undersell. I think you undersell yourself.
You're kind.
To close out here, I was wondering if I want to learn more about proteomics. I feel like I've gotten a baby master's degree in this last hour talking to you. And I listened to your podcast, which I highly recommend everyone. I'm going to pronounce it without fumbling my words. Proteomics in Proximity podcast. I like the alliteration. Yeah. What else would you recommend? Are there conferences? Are there other podcasts? Are there people that I should follow on Twitter? What do you recommend?
Yeah, great question. I think that what we try to do with the podcast is make it digestible, and we have a couple of different formats for those. Sometimes it's and I'm not the only one who hosts it. So Dale Yuzuki, Sarantis Clemitis, and me, we are 3 co-hosts on that podcast. And so we'll either just be on talking about a paper that a publication that a customer has put out, where we just, for whatever reason, wanted to talk about it before we get them on as an interview. We also do interviews. We don't, you're a pro at this. And so I will also say Patrick has been phenomenal at helping us structure our thinking about how to keep those interviews spontaneous, but also have enough structure that a guest comes in feeling comfortable on what the topics will be, so that they're not, going to go off the reservation. So thank you for that. And so those are the two primary formats. And then sometimes we'll do a live event. So like at ASHG, for example, or Hoopo or something like that, or we'll try to get a few different customers on to say something. Yeah. So I think we'd love feedback on whether that is digestible, whether it makes sense, or whether we're trying not to talk in acronyms or anything like that. So I think that's a great place. I think the Insight app is a great place. I think keeping a lookout for the UK Biobank paper that will come out soon. That will be, I'm sure, posted all over Twitter. You're welcome to follow me. I'm Taylor Lawley on Twitter. And then we're going to have a webinar that people might be interested in. By the time this podcast comes out, it's probably going to be a recording, but it'll be on the DNA Nexus website. So we're working with DNA Nexus, Chris Whelan from, again, Janssen, Ben Sun from Biogen, and Naomi from the UK Biobank are all coming on and Karsten Suri from Weill Cornell in Qatar.
Who you've had on your podcasts before.
Yes. He's such a lovely man. So we're just basically talking about the data set that is now in the UK Biobank research analysis platform, kind of what the data look like, how people are using it, how it's been prepared, how the experimental design was put together. And that will be on Wednesday, April 19th, will be the date for the actual broadcast. But again, it'll be a posted recording on the DNA Nexus website. So I think those are good resources. And then, feel free to reach out to me on Twitter or via email. I'm happy to talk to anyone about proteomics. It's very fun.
And there's 1,200 and something publications, so people can get to reading, right? If they visit your website, put it in ChatGPT.
Yeah, and I'm happy to filter it on a disease area if people are interested in that. And a lot of those publications are around the qPCR readout. So we're now seeing this momentum coming out because it was in 2020 that the NGS readout came out, that product came out. And so we're now seeing so many of these Explore publications come through peer review. And so that's really exciting as well. So I think some of the publications from the early days, Olink was officially a company in its current format in 2016, so that many of those publications, if they were trying to cast a broad net, might have had access to 1,100 proteins or 1,200 proteins in our library at that point. Now we've got around 3,000. So those same studies can be recapitulated using a broader discovery approach if needed, and if not needed, if they've got what they need, it's exciting to see folks be able to justify a discovery and an ability to move some of these things to the clinic. I'll always say this quote from Eric Topol. He has said that it takes around 20 years to get something from a discovery to the clinic, and he quotes that the stethoscope took 17 years to get into the clinic. I love that because it tells me, you know what? If there's any way to accelerate that, let's see what it's like in translating some of these protein signatures, in layering onto what we're already learning about genomics. Will that help change that? I don't know, but I like that as a benchmark to try and hit and exceed. So we'll see.
Right. Bend the curve down to 15 or 10 if you can.
Yeah. Our lifetimes are short. We need to get
20 years a long time, people. Let's get moving.
That's right. It's a career. So anyway, thanks so much, Patrick. Such a pleasure to be on. I'm still starstruck. So
there's no need to be starstruck. The best advice I think that I got about podcasting was just rule number one is make sure you have great guests, and then the rest takes care of itself. So thank you. I really appreciate all your time with this. And I look forward to maybe we can do this in a year or a year and a half, and we can update when you're at 2,000 publications and 5,000 proteins, you can catch me up again on what the latest and greatest is.
Maybe a few customers will have LDTs at that time. That would be very, very exciting. Yeah, I look forward to it anytime.
Well, thank you. And thanks everybody for listening. As always, if you like the episode, the best thing you can do, my favorite thing you can do, is just share it with a friend, somebody that you think would like it. And if you're feeling extra supportive, then go onto your favorite podcast player and hit that 5-star button. It helps other people find us, pushes us up the list. And as always, any feedback is really very much appreciated. You can find me on Twitter or email us at podcast@cynogenetics.com. Thanks a lot, and we'll see you next time.